Hi class, this is Sir Alex again. Welcome to our lesson in General Biology 1. We are finishing off our lesson 6 with lesson 6.3. It's still part of the active transport, uh, but this time we are discussing the last part of active transport, which is the vesicular transport or the bulk transport. So before we head on discussing what vesicular transport is or what bulk transport is, let's remind ourselves first what would be the goals of today's lesson. Again, it's for us basically to understand, finally finish understanding and be able to explain the mechanism of transport mechanism of the cell, particularly the active transport. More specifically, for us to be able to differentiate and explain explain the difference between exocytosis and endocytosis. So the last part of active transport is what we call the vesicular or bulk transport. It's a type of active transport wherein the cell still pushes things into its membrane or basically places things from inside to outside or from the inside of the cell to outside or from the outside environment of the cell inside its cytosol. And since it is part of active transport, you have to remember now that this type of transport requires some form of energy in order for it to happen. So if you guys look at this moving picture right here, this is actually what macrophages or basically your white blood cells do. So this is your white blood cells right here. And you had some bacteria, or it could be any cellular debris that is unwanted by the cell. And what those white blood cells do is actually engulf. That's a term. They engulf, or basically they swallow these molecular debris, or in this case, this is the bacteria that can be very dangerous or unwanted by the cell. So the whole point of vesicular or bulk transport is to actually be able to move things from the inside of the cell or from the outside of the cell inside. But this time, instead of dealing with smaller molecules or ions like what we did in the beginning part of active transport, we are dealing with more larger materials. Like in this case, we are taking in a whole bacteria. There are even instances where in the cell could take in another cell. That's what happens with the mitochondria. If you guys remember, mitochondria is believed to be a unicellular organism a long time ago, and then the cell just swallowed it using phagocytosis, which is a part of a bulk transport. So from the word itself, the word is bulk, meaning we are dealing with something bigger materials that we want to put inside of the cell or we want to transfer out of the cell. Vesicular is later we will look why it is also called the vesicular transport. The main reason that the macrophages do this is for one is to recover valuable macromolecules for the body's use. So by engulfing larger materials such as this, the macrophages can actually kind of like break it down or dissolve it, break it down into smaller macromolecules, smaller pieces that can be reused or recycled by the cell. Moreover, the white blood cells does this in order to place fragments of that pathogen. So that pathogen are basically cells, bacteria, or even viruses that can cause diseases. By white blood cells engulfing that material and breaking it down, some of the broken down materials are called fragments. These are kind of like markers that the white blood cells can place on its surface, allowing allowing for the immune cells to kind of like remember. That's how they remember various diseases that we've already encountered. Okay, if you guys kind of like probably heard how the immune system works is the vaccine works is by introducing pathogen materials inside their body, but the materials that they introduce our body, yung injection na nila sa atin or yung pinavaccine na nila sa atin are just parts of that pathogenic materials. So, halimbawa sa COVID, they take some parts of the COVID and put it that is already not dangerous to the body and introduce it to our body. Ang gagawin ng immune cell mo is to actually engulf that so that they can have this fragments on their what they call this on their surface, allowing your immune system to identify that these materials are bad. So it's basically the safe way of introducing the COVID COVID virus inside your body. So instead na introduce mo yung COVID virus in a severe way, meaning makontak mo talaga yung virus, ang ginagawa ng mga vaccine is they already take some parts of that vaccine or some parts of that virus, some parts or some identity of that virus, and introduce it to your body, allowing your immune system to be able to identify so that if ever na ma-encounter mo yung talagang virus, hindi mo na ma-experience yung buong pathogenic or dangerous experience na kayang gawin ng virus sa katawan. And this is not only true for COVID, this is actually how vaccines, this is how majority of the vaccines work. Measles, rubella, mumps, ano ba? Mm, measles, mumps, and rubellas are the type of vaccines na madalas na binibigay sa baby to introduce those dangerous pathogenic materials na in order for the baby or the kid, not baby, the kid to start their immune system to already start producing those immune cells that can antigens called antigens to protect the kid or to protect us as we grow up. So, and all of that happens by vesicular or bulk transport by basically your macrophages engulfing those materials. So that's what bulk transport is. We're basically introducing or taking in large quantities or large particles into the cell where we move it across the cell membrane. Enclosed substances to be transported in their own small globes of membrane which can then bud from or fuse with the membrane to move the substances across. So we will discuss this more later, but basically bulk transport, after engulfing that materials, after nyo swallow yon, that swallowed materials can be moved inside to our cells. Later I will show you how. Again, these reasons are for the macrophages, but in general, why do we allow this to do? Or why does the cell want to do this bulk transport? First is for the cell to allow certain materials, more specifically nutrients from the ECF, because remember the extracellular fluid, in order for the cell to take in some nutrients from the ECF and put it inside its body for its use. Also, for the cell to be able to selectively grab particles from the ECF, particles that they may have a need for. And also for them to be able to release signaling molecules for communication. So, your first two are basically taking materials from the outside towards the inside of the cell. And then the third part is for the cell to be able to actually release something into the ECF. So bulk transport work both ways. And there are two types of vesicular or bulk transport called the endocytosis. For endocytosis, we have three examples or three categories called phagocytosis, Pinocytosis, and receptor mediated. And lastly, we have exocytosis. So we will be discussing the difference between each of these bulk transport. And let's begin with endocytosis. Endocytosis, from the words itself, endo. Endo actually means internal or inside. Cytosis is the transport mechanism. So this is a transport mechanism that deals with putting things inside. So we are trying to put things inside the cell. Yun, so it's a general term for the various types of active transport that move particles inside the cell by enclosing them in a vesicle made up of plasma membrane. So how does that do that? So you have a moving picture right here, or this is your plasma membrane right here. And what the cell do is, you can think of this as your plasma membrane. This is your ECF, and this is your ICF. So this is the cytosol. So what the cell does is it uses its plasma membrane, because remember, plasma membrane is fluid, tama? It is not a stagnant. It is not a very hard structure. It flows. So the cell can, bend it. So what the cell does is it extends this part of the plasma membrane to engulf materials from the outside and pull things inside. So endocytosis is the general term because we have three types of endocytosis: phago, pino, and receptor mediated. But the process and how they do it is basically almost the same. They follow the same pattern, and there's two ways, or not two ways, there are two steps in order for them to do this. One is called by invagination. Invagination is they create a fold inward. So as you can see, this is your cytosol again. This is the ECF. As you can see, it creates a fold inside. That's called invagination. And what does that do is, as it invaginates, it creates this pocket. And this pocket is the one that can surround or engulf the materials. So ganun din yung ginawa nung moving picture kanina ng macrophages, no? It creates this invagination in order to swallow that bacteria. And then ang gagawin ng cell is the pocket, ipipinch off niya from the membrane. As you can see, the phospholipids kind of like move around in order to create a new membrane, enclosing the membrane, and then creating ito na ngayon, yung circle na to, this pocket is called a vesicle. That's why we also call this a vesicle transport. This membrane bound bubble, this pocket, is called a vesicle. And then it pinches off, and then it moves, or the cell managed to pinch off its plasma membrane by the use of specialized proteins that at this moment we do not really care for now. Just remember, it uses proteins in order to do this pinching off. Okay, and then this vesicle ngayon, it could go anywhere inside the cell where this material is needed. It could go into, the Golgi apparatus, the mitochondria, or even in the lysosomes to digest these materials. But anywhere that the cell needs these materials. And lastly, again, endocytosis includes phago, pino, and receptor mediated. Now let's talk about these three examples of endocytosis, beginning with phagocytosis. Phagocytosis basically means cell eating. So just remember that. Phagocytosis is cell eating, wherein a form of endocytosis in which large particles such as cells or molecular debris are transported into the cell. So basically, ito yung ginagawa ng macrophages. So they're basically swallowing or engulfing large particles, or they can even do that with another cell. They can swallow another cell, or they can swallow particles or debris, or basically fragments of a dead cell, something like that, and they are transported. The thing that I want you guys to remember, this is into the cell. Endocytosis is transferring things papasok sa cell. So one example of a phagocytosis is amoeba. You guys have heard of amoeba. Amoebas are basically eukaryotes, single-celled eukaryotes. And the way that amoeba find food or nutrients is by hunting and consuming their prey via phagocytosis. So their prey or their food are usually large particles. And what they do is they extend their membrane to engulf a large material. So we have a video right here. This is the amoeba right here. And these are paramecia. These are type of unicellular materials then. And as you can see, the amoeba extended its membrane and engulfed the paramecia. So as you can see, so it extends the membrane, or basically creating this invagination. So invagination, hindi lang siya biglang papasok. Invagination can also happen by extending. So no, yung membrane is reaching out. So parang humahaba sila, trying to swallow this paramecia. So, yan. So that's how amoeba works. And then they create this pocket called food vacuoles, where they dissolve the engulfed materials for nutrients or for food. Sa mga cell naman natin, it's basically the same. So the cells, once they successfully engulf the materials, they form food vacuoles. So yung parang vesicle na kanina that they can transfer, and a lot of the times the cell transfer the vesicles to the lysosomes, because lysosomes is the recycling center of the cell, wherein the lysosome will break down the particles for recycling or for use of the broken down materials. So halimbawa, yung kinain nilang materials is a nutrient that composes of carbon, oxygen, carbohydrates, and if the cell needs those macromolecules like lipids, carbohydrates, etc., i-break down yan ng lysosome in order to break it down into the most simple carbohydrates or lipids, and then the cell will be able to use that lipids or that carbohydrates or that amino acid, whatever it is. So that's how phagocytosis work. It's basically cell eating. It's putting large materials inside the cell. Now let's move with pinocytosis and receptor mediated. Sa pinocytosis naman, this is cell drinking. Cell drinking kasi not only is the cell engulfing materials, pero they actually take a small amount of the ECF. So not only are they engulfing yung materials, but they are also engulfing some part of fluid, some fluid from the extracellular fluid. So meaning, not only were they swallowing materials or engulfing materials, but they're also drinking some of the fluid from the ECF. And this actually occurs in many cell types, and it is continuous. Kasi continuous, mas mabilis siya ma-process because most of the time, the vesicle created by pinocytosis are a lot smaller than the ones created by phagocytosis. Kasi the materials being taken out by pinocytosis are usually a lot smaller. We have a moving picture right here. Yun. Okay, so these are cells right here, and this part right here is where this cell is undergoing some type of pinocytosis. And if you zoom in, so this is the fluid from the ECF, and as you can see, the membrane extends, creating this invagination, and finally creating this pocket, and sa loob ng pocket na yan, that inside that cell is the may liquid, may ECF. So this is called pinocytosis. So a difference lang, sa pinocytosis is engulfing materials with ECF, and they're usually a lot smaller, while phagocytosis is engulfing larger materials, but both happens papasok sa cells. And lastly, receptor mediated. For receptor mediated, from the word mediated, there is a help. So may help na ginagamit yung cell in order to put things inside, in order to engulf things. And yung help na yon are conducted by receptors. Receptors are type of proteins. So receptor proteins on the cell surface. So we have a lot of proteins, remember, sa surface ng cell, sa plasma membrane. And some of these proteins are transmembrane proteins. So meaning they crosses the, if you guys remember what transmembrane proteins are, it's a protein that crosses the plasma membrane. So they are exposed both in the cytosol and out on the extracellular fluid. And then these transmembrane proteins, what they actually do is they receive, that's why they're receptor, they're parang container, no? They receive the materials. They are the one that is attaching onto the materials that the cell wants to engulf. And you can find these transmembrane proteins clustered around, or they are kind of like usually nagkukumpulan sila on an area of the cell membrane called coated bits. Coated bits is a part of the cell membrane that has a lot of clathrin. Clathrin is a type of proteins that is a layer of protein that is found on the cytosol part of the cell. Para makita nyo. Ayun. So this is your plasma membrane. These are your receptors. These are your protein receptors. And as you can see, they attach onto the whatever the materials the cell wants to engulf. And they are usually found on a layer of protein called clathrin. So ito yung chain ito. This is a protein called clathrin. And this clathrin is usually the one that help create this vesicle. So ang gagawin ng receptor is they attach into the materials and then the clathrin would be triggered to do endocytosis. So kung wala naka-attach na material dun sa receptor mo, sa protein receptor mo, it will not engulf it. Pero once na na-attach yung materials na tugma dun sa receptor, dun sa shape ng receptor, the clathrin will be triggered to start the invagination, to start the creation of the pocket, and to start the endocytosis. And then it also helps pinching the, it will help pinch the membrane of creating the vesicle. So yun lang yung difference. Receptor mediated, you need the help of the receptor proteins. And they're usually found on a layer of protein called coated bits. And clathrin is just one of the most common example that you can find all over the cell. But there are other types of protein layer that can do this. Okay, so that is pinocytosis and receptor mediated. So far we've discussed endocytosis. And for summary, endocytosis is basically putting things inside the cell via phagocytosis, which is cell eating, pinocytosis, which is cell drinking, and receptor mediated, wherein the cell take materials with the help of receptor proteins. And lastly, pag-usapan natin is yung last part ng ating active transport, which is exocytosis. If endocytosis is internal cytosis, exocytosis from the word exo, which means outside. So what exocytosis is, we are actually basically releasing materials from the inside of the cell to the extracellular fluid. So if cell could take materials from the outside environment via endocytosis, it could also release materials such as signaling proteins and waste product via exocytosis. And again, exocytosis is a type of bulk transport which the cell transport materials from inside to outside. And it does this by having the vesicles fuse with the plasma membrane. So this is a picture right here. This is your plasma membrane. And you have a vesicle that starts from the inside of the cell somewhere, usually the Golgi apparatus, because that's where a lot of the proteins are created. And some of the proteins that they create are signaling proteins that needs to be released outside of the cell. So from the Golgi apparatus, that vesicle bulges off, and then it moves towards the plasma membrane and it fuses. And then it releases the material into the ECF. So this is the ECF, this is the ICF. This is the cytosol, and this is the outside environment of the cell. So many of these vesicles come from Golgi apparatus, and what happens, it could, two things could happen. Some of the vesicles can fuse. So parang nangyari dito, this vesicle fuses with the plasma membrane. So nagiging parte siya ng plasma membrane. Other times, what it can do is it fuses shortly lang. So parang ganyan lang, i-release niya, and then ang gagawin niya is babalik siya. So it will not be part of the plasma membrane. So that's what we call a kiss and run. So it was just fuse a little bit, release the material, and then go back to the Golgi apparatus. So that is exocytosis. Isa lang, tama? So I hope you guys remember that. Exocytosis is basically when we want to release materials from the inside to outside of the cell, while endocytosis is we want to take things from the outside inside the cell. So this is the end of bulk or vesicular transport, and all of these are called active transport because all of them requires energy, some form of energy, in order for the cell to transfer materials into and out of the cell. So I hope that is clear. And if you guys have any question about vesicle or bulk transport, do not hesitate to message me. Thank you so much and have a great day.